Good morning everyone. Myself Madhu Gautam, student of MSc Medical Biotechnology first year from Maharishi Dayanand University. The topic of my presentation is History and Principles of Virology, Structure and Morphology of Animals and Plants Viruses. Before we start, first we know what is virology. Virology is the study of viruses in through which focus on the following aspects of the viruses, that is their structure, classification, their evolution and their ways to infect and exploit host cells for reproduction. As we know that viruses can only live or reproduce in a living cell. Their interaction with host organisms, physiology and immunity, the disease they cause, the techniques they use to isolate and culture the viruses, and their use in research and therapy. Virology is a subfield of microbiology. What are viruses? Virus is a submicroscopic infectious agent that replicates only inside a living cells of an organism. Viruses can infect all type of life forms from animals and plants to microorganisms including bacteria and archaea. Virus particles can only be observed by an electron microscope. Parts of a viruses or virion are envelope, that is a lipoprotein membrane, capsid, that is a protein shell that surrounds and protects the nucleic acid of the viruses, genome, that is either a RNA or a DNA. Uh, Viruses have different shape, are present in different shape, size, and we can study them on the basis of their shape, size and structures. That is, viruses have an inner core of nucleic acid surrounded by a protein called an envelope, and most viruses ranges in sizes from 20 to 250 nanometers. Viruses replicate within a host cell utilizing the host cell's nucleic acid. Viral life cycle consists of 6 stages. That is, first is attachment to the host cell, penetration into the host cell, uncoating inside the host cell or multiplication of the viruses of the host cell DNA, and the assembly and the release of the virus from the host cell. The cultivation of viruses includes chicken egg culture, cell culture or animal inoculation. Now we study what is the history of virology. In 1796, Edward Jenner, that is, he used cowpox to vaccinate against smallpox. There is a story behind the vaccination of smallpox, that जेनर ने मिल्कमेड एक लेडी थी, उनको काउपॉक्स हुआ था क्योंकि उनकी काउ को काउपॉक्स था, तो उनकी उस लेडी को मिल्कमेड लेडी को काउपॉक्स था, तो उसने देखा उस लेडी को लेकिन स्मॉल पॉक्स नहीं था. मतलब उस टाइम पर उसने देखा जितने भी मिल्क मेड्स होते थे या जो भी काउ काम करते थे उनको काउपॉक्स था, लेकिन स्मॉल पॉक्स नहीं हो रहा था क्योंकि स्मॉल पॉक्स और काउपॉक्स सिमिलर एक जैसे से वायरसेस से हो रहा था. तो उसने क्या देखा, तो उसने क्या किया, जो स्मॉल पॉक्स काउ पॉक्स जिस लेडी को हुआ था उसके काउ पॉक्स के जो पस था उस लेडी से लेकर एक 8 ईयर लड़का था उसके अंदर इनोकुलेट किया था. फिर उसके बाद उस लड़के को काउपॉक्स हुआ और वो बीमार भी हुआ, लेकिन ज्यादा वो अफेक्टेड, काउपॉक्स ज्यादा इन्फेक्शियस नहीं है एज कम्पेयर टू स्मॉल पॉक्स, तो वो ठीक हो गया. लेकिन फिर कुछ टाइम बाद जेनर ने स्मॉल पॉक्स उसी सेम लड़के के अंदर स्मॉल पॉक्स के वायरस इनोकुलेट किए, लेकिन उस लड़के को, जो बच्चा था जिसके अंदर वैक्सीन इनोकुलेट की गई थी स्मॉल पॉक्स के वायरसेस, उसको स्मॉल पॉक्स नहीं हुआ. तो उस जेनर को इससे पता चला जो काउ पॉक्स के जो वायरसेस डाले थे उसने उसकी बॉडी के अंदर काउ पॉक्स के साथ साथ स्मॉल पॉक्स के अगेंस्ट भी उसने एंटीबॉडीज रेज की थी. तो जेनर वाज द फर्स्ट पर्सन to deliberately vaccinate against any infectious disease, that is to use a preparation of containing an antigenic molecule of mixtures of such molecules designed to isolate an immune response. Jenner is given the credit for vaccination, virulation, And the virulation is the practice of deliberately infecting people with smallpox to protect them from the worst type of diseases. As Edward Jenner discovers the vaccine against smallpox, he is known as the father of immunology. In 1885, Louis Pasteur experimented with the rabies vaccination using the term viruses to describe the agents of rabies. Although Pasteur did not discriminate between viruses and other infectious agents, Louis Pasteur originated the term virus and the vaccination in honor of Jenner and developed the scientific basis for the Jenner's experimental approach to vaccination. In 1886, John Buist, a Scottish pathologist, stained the lymph skin lesions of a smallpox patient and showed elementary bodies, which he thought were the spores of micrococci. These were in fact smallpox virus particles just large enough to see with light microscope. In 1892, Dmitry Ivanovsky described the first filterable infectious agent through tobacco mosaic virus that is smaller than any known bacteria. He was the first person to discriminate between virus and other infectious agent, although he was not fully aware of the significance of his finding. In 1889, Martinus Bijnik extended Ivanovsky's work with TMV and formed first concept of virus contagium vivium fluidium, which means soluble living germ, and was the first person developed the concept of viruses as a distinct entity. By the work he done, he is known as the father of virology. In 1900, Walter Reed demonstrated that yellow fever is spread by mosquitoes, and he and his workers were the first to show that viruses could be spread by insect vectors such as mosquitoes. In 1908, Karl Landsteiner and Erwin Popper proved that poliomyelitis was caused by a virus, and Landsteiner and Popper were the first to prove that viruses could infect humans as well as animals. In 1911, Francis Python Rose demonstrated that a virus, that is Rose sarcoma virus, can cause cancer in chickens. He is awarded with Nobel Prize in 1966 for his work. Frederick Twart discovered viruses infecting bacteria, and in 1917, Dlx D. Harrell independently discovered viruses of bacteria and coins the term bacteriophage. In 1935, Wendell Stanley crystallized TMV, that is tobacco mosaic virus, and showed that it remains infectious, and it is first step towards describing the molecular structures of any viruses. And Stanley got awarded with Nobel Prize in 1951. In 1938, Max Thiller developed a live attendant vaccine against yellow fever and got Nobel Prize in 1951. In 1939, Emory Ellis and Max Delbruck established the concept of one-step virus growth cycle, essential to understanding of virus replication. This work laid the basis for the understanding of viruses replication, that virus particles do not grow but are instead assembled from performed components. In 1940, Helmut Ruska used an electron microscope to take the first pictures of virus particles. In 1941, George Hist demonstrated that influenza virus agglutinates red blood cells, and this was the first rapid quantitative method for measuring eukaryotic viruses. In 1945, Salvador Luria and Alfred Hershey demonstrate that bacteriophages mutate and got Nobel Prize in 1969. This works proves that similar genetic mechanism operated in viruses as in cellular organisms and lays the basis for understanding of antigenic variations in viruses. In 1949, John Anders and Thomas Weller and Frederick Robbins were able to grow poliovirus in vitro using human tissue culture, and they got Nobel Prize in 1954. This development led to the isolation of many new viruses in tissue cultures. In 1950, Andrei Lvov, Louis Sminovich, and Niels discovered isogenetic bacteriophages in Bacillus megaterium, irradiated with ultraviolet light, and coined the term prophase. And they got Nobel Prize for their work in 1965. Also in 1950, the WHO proposed a program to eradicate smallpox from America, and this was achieved in 8 years. In 1952, Renato Delbuco showed that animal viruses can form plagues in a similar way to bacteriophages. Delbuco works allowed rapid quantitation of animal viruses using assays which had only previously been possible with bacteriophages, and he awarded with Nobel Prize in 1975. Alfred Hershey and Martha Chase demonstrate that DNA was the genetic material of bacteriophage, although the initial evidence for DNA as the molecular basis of genetic inheritance was discovered using a bacteriophage. In 1957, Heinz Frankel Conard and R.C. Williams showed that when mixtures of purified tobacco mosaic virus RNA and code protein were incubated together, virus particles formed spontaneously. And in 1957, Alice Isaacs and Jean Lindemann discovered interferons. In 1966, Baruch Blumberg discovered hepatitis B virus and got Nobel Prize for his work for his discovery in 1976. He went on to develop the first vaccine against the HBV, that is hepatitis B virus, considered by some to be first vaccine against cancer because of the strong association of hepatitis B with liver cancer. In 1970, Howard Tenem and David Baltimore independently discovered reverse transcriptase in retroviruses, and they got Nobel Prize in 1975 for their work. In 1973, Peter Daugherty and Rolf demonstrate the basis of antigenic recognition by the cellular immune system and awarded with Nobel Prize in 1996. In 1973, J. Michael Bishop and Harold Varmus discovered that the oncogenes from Rous sarcoma virus is also found in the cells of normal animals, including humans. Proto-oncogenes are essential for normal development but can become cancer genes when cellular regulators are damaged or modified, example by virus transduction. And they got Nobel Prize in 1989. In 1981, Yorio Hinuma and colleagues isolated human T-cell leukemia virus, that is HLTV, from the patient with adult T-cell leukemia. In 1983, Montagnier and Robert Gallo announced the discovery of human immunodeficiency virus, that is HIV, the causative agent of AIDS, and it only 2 years since the start of it only 2 years since the start of the AIDS epidemic disease, the agent responsible for this disease has been identified. In 1986, Roger Bechet and Rob Frele and colleagues demonstrated that tobacco plants transformed with the gene for the code protein of tobacco mosaic virus and are resistant to TMV infection. In 1990, first approved human gene therapy procedure was carried out on a child with severe combined immunodeficiency disease using a retrovirus vector. In 1999, nucleotide sequence of the largest viral genome yet known completed, that is Paramecium bursaria cholera virus 1. In 2001, the complete nucleotide sequence of human genome is published. Now we know about the structure and morphology of animals and plant viruses. Classification of viruses. Viruses are small obligate intracellular parasites which by definition contain either RNA or DNA genome surrounded by a protective virus-coated protein coat. The following properties has been used as the basis for the classification of viruses. Virion morphology including size, shape, type of symmetry and presence or absence of the membranes. Virus genome properties. On the basis of virus genome properties, that is include type of nucleic acid, that is DNA or RNA, size of genome in kilobases or kilobase pairs, and strandedness, that is single stranded or double stranded, whether linear or circular, or sense, that is positive, negative or ambisense, segments, number and size, nucleotide sequences, that is G plus C content, and the presence of spatial features, that is repetitive elements, isomerization, 5' terminal cap and 3' terminal poly A tracts. Genome organization and replication. On the basis of genome organization and replication, including gene order, number and position of open reading frames, a strategy of replication, that is pattern of transcription translation, and cellular sites, that is accumulation of proteins, viral assembly and viral releases. Are on the basis of virus protein properties including number, size and functional activities of structural and non-structural proteins, amino acid sequences, modifications, that is glycosylation and phosphorylation, and spatial functional activities, that is transcriptase, reverse transcriptase, neuraminidase and fusion activities. On the basis of antigenic properties and physiochemical properties of the viruses, including molecular mass, beyond density, pH stability, thermostability, and susceptibility to physical and chemical agents, especially either detergents, ether and detergents. Biological properties including natural host range, mode of transmission, vector relationship, pathogenicity, and tissue tropism and pathology. On the basis of genetic material present in the viruses, they are classified as a virus consists of genetic information, either DNA or RNA, coated by a protein. Accordingly, they are classified as DNA viruses and RNA viruses. The nucleic acid may be single or double-stranded, circular or linear, segmented or unsegmented. DNA viruses, as their name implies, DNA viruses use DNA as their genetic material. Some common example of DNA viruses are papovirus, papillomavirus, and herpes virus. DNA viruses can affect both humans and animals and can range from causing benign symptoms to posing very serious health disease. RNA viruses, the viruses that possess RNA as their genetic material are called RNA viruses. Examples are rotavirus, poliovirus, yellow fever virus, dengue virus, hepatitis C virus, measles virus, rabies virus, influenza virus, and Ebola viruses are examples of RNA viruses. DNA RNA viruses are the RNA tumor viruses called leucoviruses, and Rous viruses unusually contain both DNA and RNA as genetic material. Classification of viruses on the basis of structure and their symmetry, that is helical or cylindrical viruses. The helical viruses are elongated, rod shaped, and rigid or flexible in nature. Their capsid is a hollow cylinder with a helical structure. Capsid consists of monomers arranged helically in a rotational axis, and helical or cylindrical symmetry viruses examples are tobacco mosaic virus, bacteriophage M13, and influenza viruses consist of the helical viruses consist of numerous identical capomers arranged in a helix form. Polyhedral isohedral viruses. Polyhedral structures has three possible symmetries such as tetrahedral, octahedral, and icosahedral. Icosahedral symmetry, many viruses have spherical or cubical or polygonal shapes, which is basically icosahedral or 20-sided. That is, examples are bacteriophage, turnip yellow mosaic viruses. The viruses are more or less spherical. Therefore, icosahedral symmetry is best one for the packaging and bonding of the subunits. The capomers of each phase form an equatorial triangle and 12 intercepting points or corners. They consist naked capsid, example adenovirus, or envelope viruses, and that is herpes simplex virus. Complex viruses, the viruses which have undefinable capsids or have the capsids with additional structures are called complex viruses. Capsids not clearly identified in the complex viruses, examples are vaccine viruses, pox viruses such as vaccina or cowpox. Rabies virus is a bullet-shaped, etc. Capsids to which some other structures are attached, that is, an example is bacteriophages, are complexes viruses. Some viruses like influenza virus and mump viruses are surrounded by a 100 to 150 angstrom thick spiked membrane. These membrane consist of lipid bilayer or plasma membrane. These are the helical viruses that are helix in small capomers are arranged in a helix form, and the center is a hollow. This is the example or picture of a icosahedral virus, and this type of viruses are known as complex viruses, that extra spike-like structures are attached to the main body of the virus. Now we classify viruses on the basis of their capsid structures. Viruses with naked icosahedrals viruses whose capsids are not covered by an envelope and they show icosahedral symmetry are known naked icosahedral viruses. Examples are hepatitis A virus or poliovirus. Enveloped icosahedral viruses. Viruses whose capsids are covered by an envelope and icosahedral in symmetry are called enveloped icosahedral. Examples are Epstein-Barr virus, herpes simplex virus, rubella virus, and yellow fever virus or HIV-1. Enveloped helical viruses, viruses whose capsids are covered by an envelope and they show helical symmetry are called enveloped helical viruses, and the examples are influenza virus, mumps virus, measles virus, and rabies virus. Naked helical viruses are viruses whose capsids are not covered by an envelope and they show helical symmetry are called naked helical viruses. And examples are tobacco mosaic virus. Complex with many proteins, these type of viruses have some combinations of icosahedral and helical capsid structures and additional structures attached to the capsid, and examples are herpes virus, smallpox virus, hepatitis B virus, and T4 bacteriophage. On the basis of number of strands, the presence of number of strands, the viruses are classified as double-stranded DNA viruses. If it is found in pox viruses, the bacteriophages T2, T4, T6, T3, T7, and lambda herpes viruses or adenoviruses. Single-stranded DNA viruses, it is found in bacteriophage X and 74 bacteriophage. Double-stranded viruses are found within the viral capsids in the retroviruses of animals and in the wound tumor viruses and rice draft viruses of plants. Single-stranded RNA viruses found in most of the RNA viruses. Examples are tobacco mosaic virus, influenza virus, poliomyelitis, and bacteriophage MS2 viruses. On the basis of type of host, viruses are classified on the basis of the type of host. They are classified as animal viruses, plant viruses, and bacteriophages. These are animal viruses are the viruses which infect and live inside the animal cells, including men, are called animal viruses. Examples are influenza virus, rabies virus, mumps virus, polio virus, etc. Their genetic material is RNA or DNA. The plant viruses are the viruses that infect plants are called plant viruses, and their genetic material is RNA, which remains enclosed in the protein coat. Some plant viruses are tobacco mosaic virus, potato virus, beet yellow virus, and turnip yellow virus, etc. Bacteriophages. Viruses which infect bacterial cells are known as bacteriophage or bacteria eaters. They contain DNA as genetic material. There are many varieties of bacteriophage. Usually, each kind of bacteriophage will attack only one species of or only one strain of bacteria. According to the Baltimore classification of viruses, they are grouped into 7 groups. That is, first group viruses have double-stranded DNA. In second group, single-stranded DNA viruses. In third group, double-stranded RNA viruses. In group 4, positive sense single-stranded RNA viruses. And in group 5, negative sense single-stranded RNA viruses. In group 6, viruses are single-stranded RNA reverse transcribing viruses, and in group 7, double-stranded DNA reverse transcribing viruses are included. Now we studied each group. Group 1, their mRNA is produced by transcription in much the same way as with cellular DNA. That is, the mRNA is synthesized on double-stranded DNA genome template. Example of group 1 viruses are adenoviruses, herpes viruses, papoviruses, and pox virus. Adenoviruses. Virions are naked, not enveloped, and that is 90 to 90. Adenoviruses are 90 to 100 nanometer in diameters and have icosahedral symmetry. The pendant fibers with a globular head are involved in the process of attachment of the virus to the host cell. These are the pendant globular structures. Hexons consist of protein 2 with a central core involved in the stabilization of assembly of particles. Disease caused by adenoviruses are respiratory diseases, conjunctival pneumonia, acute hemorrhagic cystitis, and acute gastroenteritis. Herpes virus. The nucleocapsid is spherical, icosahedral, and 100 nanometer in diameters, enclosed in a glycoprotein lipid envelope. Glycoproteins are visible as spikes that are projected from the surface, and the nucleic acid is linear double-stranded DNA, which is converted into circular DNA molecule upon slight digestion. Disease caused by herpes viruses are herpes simplex 1, that is cold sores, herpes simplex 2, genital sores, and versiloviruses, that is chickenpox or shingles. Papoviruses. Capsid is of 55 nanometer in diameter, naked, icosahedral. Virion consists of DNA and proteins, and capsid is made up of up to 72 capsomers. Virus enters the cells and migrate to the nucleus for the replication. Disease caused by the papovirus are papillomavirus, that causes cervical cancer and human warts, and polymavirus, that causes tumor in mice. And papovirus looks like this. Pox virus. Pox virus are brick shaped, with 240 to 300 in nanometer, double-stranded DNA genome, and naturally associated with enzymes. Virions are enveloped. Viral multiplication is started by viral transcriptase. The viral components are synthesized and assembled in the cytoplasm of the host cell. Pathogenesis by poxviruses are human monkeypox and smallpox. Group 2. Group 2 viruses have single-stranded DNA. An example is of paravovirus, as their genome have single-stranded DNA. They convert their single-stranded genome into a double-stranded DNA intermediate before transcription to mRNA can occur. Parvovirus are linear, average genome size of 500 nucleotides, smallest virus with 18 to 28 nanometer in diameter. Viral capsid made up of 2 to 4 proteins from naked icosahedral structures. Disease caused by parvoviruses are human parvovirus B19, erythema infectiosum, and joint disease syndrome. Group 3 viruses use DNA, double-stranded RNA as their genome, and the strands separate, and one of them is used as template for the generation of mRNA using the RNA-dependent RNA polymerase encoded by the virus. And the examples are coronavirus, picornavirus, and toga virus. Coronavirus viruses are enveloped, helical, and 60 to 200 nanometer in diameter, and genome size are of 60 to 21 kb. It replicates in the cytoplasm, and disease caused by this virus are respiratory syndrome, severe acute respiratory syndrome. The coronavirus looks like this. Picornavirus. Virus are naked icosahedral. Capsid is composed of 64 copies of 4 structural proteins, and viral genome are linear and 7.1 to 8.9 kb in size. And disease caused by picornaviruses are poliovirus, polio, common cold that is caused by rhinovirus, and hepatitis A virus which causes hepatitis A. And they looks like this. Toga virus. They have enveloped consist of 80 trimer spikes. The spikes consist of glycoproteins that help in attachment of protein to receptors of the host cell membranes. An example of toga viruses are alpha virus, Eastern equine encephalitis virus, yellow fever virus, and rubella virus. And it looks like this. Group 4 viruses. Group 4 viruses have single-stranded RNA as their genome with a positive polarity or positive sense. Positive polarity means that the genomic RNA can serve directly as mRNA, and intermediates of double-stranded RNA called replicative intermediates are made in the process of copying the genomic RNA. Multiple full-length RNA strands of negative polarity that are complementary to the positive-stranded genomic RNA are formed from these intermediates, which may then serve as the templates for the production of RNA with positive polarity, including both full-length genomic RNA and a shorter viral mRNAs. Example of group 4 viruses are rabedovirus and orthomyxovirus. Rhabdoviruses are bullet-shaped with one end flattened like this. This end is flattened and the upper one is a bullet shape. Rhabdo is a Greek word which means a rod, but the term rod is not accurate to its morphology. Rhabdoviruses are found in vertebrates, invertebrates, and plant. The example of rhabdovirus are rabies virus, mumps virus, vascular stomatitis virus, and potato yellow draft virus. Vascular stomatitis virus, that is VSV, is the most extensively studied rhabdovirus. It is a mild pathogen to cattle, and vesicular stomatitis virus affects the central nervous system, so host parasite do abnormal behavior. VSV contain a nucleotide capsid which is enveloped on its outer surface, consists of numerous spikes of glycoproteins on the inner surface of the membrane protein. Group 5 viruses. Group 5 viruses contains single-stranded genome with negative polarity, meaning that their sequence is complementary to the mRNA. As with group 4 viruses, double-stranded RNA intermediates are used to make copies of the genome and produce mRNA. In this case, the negative stranded genome can be converted directly to the mRNA. Additionally, full-length positive RNA strands are made to serve as templates for the production of the negative stranded genome. Example of group 5 viruses are revoviruses. Revoviruses or revoviridae have wide host range including vertebrates, invertebrates, plants, protists, and fungi. They lack lipid envelopes and package their genome of discrete double-stranded segments of RNA within the multiple layered capsids. Lack of lipid envelope has allowed 3D structures of these large complex viruses, which are nearly 60 to 100 nanometer in diameter, to be obtained. Retroviruses can affect gastrointestinal system such as rotaviruses and respiratory tracts, and retroviridi looks like this. Group 6 viruses. Group 6 viruses have diploid, that is two copies of single-stranded RNA genomes that must be converted using the enzyme reverse transcriptase to double-stranded DNA. The double-stranded DNA is then transported to the nucleus of the host cell and inserted into the host genome. Then mRNA can be produced by transcription of the viral DNA that was integrated into the host genome. Example of group 6 viruses are retroviruses. Retroviruses or retrovirids have genome of plus sense RNA. Capsid is made up of protein. Envelope is a lipid glycoprotein. Enzymes that retroviruses have are reverse transcriptase, integrase enzyme, and protease enzyme. An example of retroviruses are HTLV1, that is human T cell lymphotrophic virus. Retroviruses are those viruses which cause HIV in the humans. Structure of a retrovirus is like this. Group 7 viruses have partial double-stranded genome and makes a single-stranded intermediates that act as mRNA but are also converted back into double-stranded DNA genome, but are also converted back into double-stranded genomes by reverse transcriptase and necessary for genome replication. Example of group 7 viruses are herpesvirus. And herpesvirus looks like this. This is the structure of herpesvirus. Herpesvirus, as the name is based on hepato, liver, and trophic, replicates and cause infection in the liver, and are DNA viruses as they have DNA in as its their nucleic acid. The herpesvirus family has the smallest genome of all replicating component animals DNA viruses. The single most important member of the family is hepatitis B virus, HBV. HBV exhibits an extremely limited host range, infecting only chimpanzees and humans. The Hepadina family of the viruses consists of 5 viruses. One human virus, that is hepatitis B, and 4 animal viruses, that is woodchuck hepatitis virus, ground squirrel hepatitis virus, Peking duck hepatitis virus, and other avian hepatitis virus. These 5 viruses are classified together for the following reasons. They all are enveloped. They all contain polymerase that can repair the viral DNA genome during replication, and they produce lipoprotein containing envelope proteins. They infect species that are closely related to their natural host, as they have the narrow host range. As the family Hepadina or hepatotrophic DNA viruses implies, they can produce chronic infections in liver cells. As stated above, we will focus only on the human virus in the family hepatitis B. Animal viruses. Animal viruses are self-replicating intracellular parasites that completely rely on host animals for reproduction. They use host cellular components to replicate and then leaves the host cells to infect other cells. Animal Animal viruses mainly infects animal cells and cause different diseases in animals including man. Contains genetic material in the form of either DNA or RNA. The protein coat or capsid of animal viruses is surrounded by an envelope. Polymyelitis is most extensively studied RNA containing animal virus. Common viral diseases of animals are common cold, influenza, mumps, chickenpox, smallpox, polio, etc. Pox virus is the largest virus among the animal viruses. Plant viruses. Plant viruses are viruses that affect plants like all other viruses. Plant viruses, like all other viruses, are obligate intracellular parasites that do not have the molecular machinery to replicate without a pathogenic to higher plants. Example of plant viruses are tobacco mosaic virus, barley yellow dwarf virus, tomato spotted wilt virus, tomato yellow leaf curl virus, cucumber mosaic virus, cauliflower mosaic virus, etc. All plant viruses consist of ribonucleoprotein in their organization except few exceptions like cauliflower mosaic virus DNA. Important plant viruses are tobacco rattle virus, tobacco mosaic virus, potato virus, turnip yellow virus, etc. Citrus tristeza is longest among the plant viruses. Plant viruses mainly spread by insects such as aphids, leaf hoppers, and beetles. Tobacco mosaic virus is the most extensively studied plant virus. It was discovered by Ivanovsky and crystallized by Stanley and plants including tomatoes, peppers, cucumbers, and tobaccos may all be infected by the tobacco mosaic virus. TMV is a rod-shaped RNA single-stranded virus and are cigarette-like shaped. Plant diseases produced disease in plants disease produce symptoms such as ring spots, mosaic pattern development, leaf yellowing and distortion and deformed growth. Some plant viruses are not limited to one particular plant host, may infect different varieties of plants. Now we see some now we know about some viruses, plant viruses such as tobacco mosaic virus. Classification of tobacco mosaic viruses families Virgaviridae, genus is Tobamovirus, and species are tobacco mosaic virus. Structures of tobacco mosaic virus is rigid helical rods and single-stranded RNA, extremely stable structure, 95% protein and nearly 5% nucleic acid, that is RNA. 2,130 copies of single 17 kilodalton code protein and 300 nanometer long and 18 nanometer in diameter. Rod-like appearance, and there are 3 RNA nucleotide monomers. The first plant virus discovered and attacked members of nightshade or Solanaceae family including tobacco, pepper, potato, tomato, eggplant, cucumber and petunia. Spreads through the entry into breaks of the cell walls caused by insects or other physical damage. Tobacco mosaic virus looks like this. Barley yellow dwarf virus infects several grains and staple crops including wheat, bajra, etc. Aphids primarily spread the virus. This virus causes discoloration of leaves and the tips of the plants, reduce photosynthesis, stunts growth and decrease production of seed grains. These yellow leaves are caused due to this virus. Now, thank you.